Hello, everyone. I have the distinct pleasure to introduce Krutika Patidar is a PhD student in my laboratory. She earned her B.Tech in chemical engineering from VIT University in India in 2017. And then she joined UB as a master's student in chemical engineering. She first was in Johannes Hockman's lab and successfully completed her master's degree. And as I was transitioning here and COVID times, she reached out to me to express interest and becoming one of my first UB PhD students, and it's been a fantastic experience. She has worked with me since fall of 2020, so she was in the department before I arrived in January 2021. While she's been here, she's been super productive. She also did a 6-month co-op at Sanofi Pharmaceuticals in Boston last summer and fall. She plans to graduate over winter this year for the February conferral date and has multiple publications, including some from that co-op experience, as well as some work that's just our lab and some collaborators. So she's going to talk about her main core PhD project today. She's also been super involved in leadership with the Graduate Society of Women Engineers here at UB, in addition to pioneering some work in our lab, being the first student to adopt this logic-based ordinary differential equations framework. So it'll be a little bit different than what some of the other people in my lab, or maybe you've seen in some other mathematical kind of talks, but it'll spread between computational mathematics and bio. So hopefully there's something in the audience for everybody here, to maybe a little on materials too. But all right, so Krutika.
Thank you for that lovely introduction. All right, let's dive in. So diabetes is a long-lasting health condition that currently affects 1 in 11 adults, and among 1 in 3 patients with diabetes develop diabetic kidney disease. There is currently a high risk of or increasing trend in the diabetic kidney disease cases just in the 7 major markets, and these cases are over 82 million as of 2023. But this trend is increasing, and these numbers are even higher across the world. There's also a high risk of progression of diabetic kidney disease to chronic kidney disease, as well as end-stage renal failure. And this is one of the highest causes of deaths in the diabetic kidney disease patients. So diabetes impairs glucose control and metabolism in different cells, including immune cells like macrophages. We are interested in understanding how glucose and inflammation lead to the development of diabetic kidney disease through changes in the immune cell microenvironment, and how this may cause metabolic reprogramming within these cells and affect them. So for instance, activation of a high inflammatory stimulus can trigger an activation of immune cell receptors within macrophages, which is shown through this broader image of metabolic pathways and cell survival maintenance pathways within macrophages. This activation of inflammatory cells can trigger an increased glucose uptake through the glucose receptors within these cells and impair their glycolytic pathways, which can lead these macrophages to switch to and remain in an inflammatory phenotype, often triggered through an increased transcription and translation of inflammatory cytokines. So the circulating immune cells or macrophages with an imbalanced inflammatory phenotype can infiltrate the kidney. Kidneys are a multitasking organ that are responsible for removing toxins and waste from the blood, but also regulating blood flow, volume, and pressure. And once the diabetic kidney disease develops, these imbalanced inflammatory macrophages can target the glomerulus within the kidney. So the glomerulus is a complex network of capillaries within the functional unit of the kidney that is responsible for maintaining the structure and function of the kidney. It is also a specialized structure that is defined by different size-selective cellular layers, including the fenestrated endothelial cell layer, the mesangium, basement membrane, and podocytes. Each of these size-selective layers can undergo some sort of signaling dysregulation as well as structural changes within them as the diabetic kidney disease progresses. So as compared to the healthy glomerulus, diabetic kidney disease affected glomerulus will undergo structural changes within the cellular layers. This mainly affects their size, porosity, and arrangement within the glomerulus. For instance, the basement membrane may thicken, there may be mesangial expansion, there may be loss of these fenestrations or pores in the endothelial cell layer, as well as loss of their size-selective and jar-selective properties. And then the podocytes may also undergo injury and loss. So we are interested in understanding how glucose-mediated inflammation in the macrophages activates the glomerular endothelial cell layer and causes signaling dysregulation. And we're interested in looking at two main events, that is changes in the intercellular endothelial barrier, which is the barrier maintained by different junction proteins at the surface of the endothelial cell layer, and how affecting this barrier can increase permeability or cause leaky endothelial cells. And then another thing we're interested in is how signaling dysregulation will affect the size and arrangement of these pores or these fenestrations in the endothelial cell layer, which are essentially supporting the size-selective properties of the endothelium. Yet, there are not many mechanisms that are defined on how these endothelial cell layers are regulated and their role in diabetic kidney disease. So we leverage mathematical modeling to understand the mechanisms of action that trigger this glomerular endothelial cell dysfunction, especially in the early stages of DKD. So mathematical modeling allows us to represent any biological system in hopes to understand it as well as quantify it. So here, any mathematical model may range in complexity based on the research question at hand. So we may leverage a mathematical model that looks at a holistic multi-scale understanding of different organs, cells, molecular networks, or genes within the human body. Or we may use a fit-for-purpose mathematical model that looks at a real system and uses experimental data to understand it. Now we could leverage different types of data modalities like biological, physiological, or pharmacological to build these mathematical models. And we could also use different approaches ranging from logic-based, probability-based, equation-based, as well as now popular machine learning models as well. And then once the model has been formulated, we may use this optimized model to augment our understanding of the real system as well as suggest improvements. So the workflow of this study involves first understanding the biological system of interest and the scale of interest. So we are focused on understanding intracellular and intercellular crosstalk between macrophages and glomerular endothelial cells. For this purpose, we look at 2D co-culture in vitro systems that study these two cells. And then we collect data from published studies that report changes in the protein concentrations, mRNA levels, as well as use other specialized assays to measure oxidative stress or nitrite concentrations as they change as the disease progresses. The next question or next stage in this model development process is to identify what mechanisms or pathways are most relevant in understanding the early stages of DKD. As DKD is currently affected by many different complex and interlinked processes, this was done by going through a manual survey through pathway databases, information in literature, as well as experimental studies, and putting together a protein-protein interaction network between macrophages and glomerular endothelial cells that is able to capture some relevant disease mediators and biomarkers like cytokine expression, oxidative stress, as well as angiogenic factors. And then we model the interaction network using a Netflix-derived logic-based ordinary differential equation. Netflix was actually proposed by Dr. Sossaman's group and is a technique that we're currently adapting to build this model. And then the model was simulated, fitted, and validated using the in vitro data that we have collected. So this is the protein-protein interaction network that we put together that focuses on the interaction between the macrophages and endothelial cells. It is stimulated by a high glucose and a high inflammatory stimulus, LPS. We also consider glucose-mediated activation of advanced glycation end products and their uptake by their receptors in different cells, as this phenomenon is strongly associated with diabetes progression. We include the activation of inflammatory stimulus, such as LPS in this case, and the downstream response triggered by different cell survival and maintenance pathways within the macrophages, and how it may lead to an overexpression of inflammatory cytokines, as well as oxidative stress, and the expression of vascular endothelial growth factor. So in our model, a vascular endothelial growth factor essentially activates the glomerular endothelial cells and causes changes in the nitric oxide levels within the endothelial cells, increased oxidative stress, and ultimately affects the intercellular junction integrity between two or more endothelial cells. So briefly, the mechanisms of action and the pathways that we put together in this network are not really easy to parameterize for a mathematical model. So for this reason, we consider a logic-based ordinary differential equation model because we only have limited knowledge about the rate constants or kinetics to define these interactions. So we define each of these interactions using a normalized Hill function, depending on whether it's an activation function shown as a black arrow or a inhibition function shown as a red edge. And then the protein activity or the activity of each node in the network is measured as a normalized value between 0 and 1, which is computed as an ordinary differential equation that essentially has a source term and a degradation term, where the source term is computed by applying a logic-based gate on the activation or inhibition interaction leading to the activity of that protein. So this model is defined by a set of reaction or species parameters, which basically control the strength of each interaction, as well as the time it takes for each protein to get activated or inhibited. And then before we go into results, I just want to briefly go over some of the model development steps that we considered to build a robust biological model. So we first conducted a structural observability analysis of this dynamical system and checked for whether the parameters of the model were identifiable. We then computed what parameters in the model were most sensitive through a global sensitivity analysis that gave us a smaller parameter subspace to be optimized and fit to the observed data. And then we optimized those sensitive parameters using a nonlinear least squares optimization technique, and also quantified uncertainty in these estimated parameters, as well as the prediction observed from these fitted parameters, which was computed as a 95% credible interval. So on the left, I'm showing you the model fitting results that were the simulations were fitted for a treatment condition similar to the co-culture experiment, where only high glucose stimulus was provided and no inflammatory stimulus was present. So in this case, the protein activities, as you can see, are normalized values between 0 and 1, where a normalized value of 0 represents protein concentration at baseline, or normalized value of the protein concentration at baseline, and the value of 1 would represent a maximum upregulation or activation of that protein. So as you can see, we've fitted our simulations to this data and also checked for uncertainty in these predictions using the 95% credible intervals. But we also used separate in vitro data that was used to validate this specific treatment case. And we found that under high glucose conditions alone, we saw overexpression of inflammatory cytokines, as well as oxidative stress, and then a downregulation of vascular endothelial growth factor and a subsequent inactivation of nitric oxide, which is one of the critical events that happens in the early stages of diabetic kidney disease development. In the next case that we recreated was simulating this model under a high inflammatory stimulus like LPS and a normal glucose levels or normal glucose conditions. So besides seeing an increased expression of inflammatory cytokines and oxidative stress, we also saw an increase in vascular endothelial growth factor and a subsequent increase in nitric oxide activity, which is overexpressed in the endothelial cells. And nitric oxide is a vasodilator, so it could be indirectly related with leaky endothelial cells as well as increased glomerular endothelial cell permeability. These results were also validated using experimental data that was not used in fitting. Interestingly, we also were able to validate the expression of VE cadrins or phosphorylated VE cadrins, which are the junction proteins present at the surface of the endothelial cells. And phosphorylation of these adrin junctions can cause opening of the junction as well as increase in gap between two or more endothelial cells. Lastly, we simulated our model for presence of both high inflammatory stimulus and high glucose expression as was done in the in vitro experiment and fitted those results. And then and validated using available data that was not used in fitting. But the only difference we observe is that we see enhanced expression and early activation of pro-inflammatory cytokine because both of these treatment conditions are responsible for overexpression and upregulation of these proteins. So with fitting our model to experimental data, we were able to also validate the interactions that we put together and the assumptions that we made to create this network in the first place. But we really want to do is identify some potential pathways or treatment strategies that could intervene this development of diabetic kidney disease or control the changes that are observed in glomerular endothelial cells. So the first in silico experiment that we did was locally inhibiting each of the reactions within the network or these interactions in the network, and also locally inhibited each protein or node in the network to see how this may affect the expression of downstream proteins that are not directly connected with each other. So in this first test, we found the influential interactions shown in yellow and influential or potential targets shown in yellow, which could be either tested experimentally to check for their effect on the disease development, or they could be modeled as a quantitative model and could be tested as a potential treatment strategy. The next in silico test that we did was controlling the strength of glucose. As it is a normalized Hill equation-based ODE model, the way we control our glucose levels is by changing sort of the weight of this glucose in the model. But what it would be based on real experiments would be controlling the glucose concentration from its maximum levels. So reducing glucose levels by 50 to 100%, we saw that it was able to better regulate the expression of inflammatory cytokines, oxidative stress, as well as some tyrosine kinase activity like PLC gamma, as PLC gamma can be directly or indirectly related with the changes in the interstellar barrier integrity, as well as changes in the permeability in endothelial cells. And then we also checked how some of our most influential reaction pathways, as well as proteins, would be instrumental in reversing the increased gap width between endothelial cells. This is caused by the phosphorylation of different adherens junctions. And the model simulated sort of reversing this increased gap width when each of these reactions and proteins were inhibited. So mostly we observed that VEGF-mediated activation of VEGF receptor 1, as well as downstream activation of the pathway by PLC gamma was responsible in controlling this gap width between two or more endothelial cells. So in conclusion, we've shown how inflammation and interactions between glomerular endothelial cells and macrophages are important in diabetic kidney disease. We've shown how logic-based ODs are useful in exploring protein response and pathways, especially when mechanistic knowledge and experimental validation for this is limited. And then this modeling technique is suitable to be integrated with other types of models, as well as be integrated with some other commercially available tools that analyze the dynamical system. So some other groups have integrated this model with agent-based models, as well as pharmacokinetic and pharmacodynamic models to understand, to bring in the effect of pharmacology on in these disease models. And then in this effort, we have tried to identify some testable hypotheses that can be beneficial in controlling cellular dysfunction in diabetic kidney disease. So there are existing drugs that do regulate VEGF receptor 1, calcium, and transcription factor activity. So maybe using them to see how they may affect glomerular endothelial dysfunction in diabetic kidney disease would be useful. And lastly, this is some of the ongoing and future work to expand this model is translating the existing model that we fitted to in vitro data to study mechanical changes within the cells. So by that I mean is the structural changes observed in the fenestrations or pores of the glomerular endothelial cell layer, looking at the effect of systemic glucose exposure and how it may cause signaling dysregulation, and also figuring out pathways that may connect signaling dysregulation with mechanical changes. And then also trying to understand disease intervention strategies in vivo or in diabetic mice, how different time or concentrations of glucose may affect the expression of these proteins, as well as looking at individual or combined treatment strategies by performing inhibition experiments on this network would help us identify some new treatments for diabetic kidney disease. And with that, I'd like to thank my advisor, Dr. Ashlee for Versypt, for her guidance and support, as well as my lab members who I've worked with or have been supportive as well. I would also like to thank the funding sources. NSF and NIH both supported this work. And finally, the department for the opportunity to speak today.
I think we have time for a few questions for Krutika. Oh.
Yeah.
I was wondering if inhibiting inflammation would be a way to treat diabetes, because it doesn't cure diabetes, right? Because
Right.
those cells are damaged and they are trying to have their inflammation to repair.
Right.
But when you inhibit inflammation, then they are going to look better, the pore size goes smaller, and the kidney is going to be improved, but they are still damaged, right?
Yeah. So I think what I understand is that if you were able to find that there is changes at a cellular scale, changes happening in these diabetic patients, and diabetic kidney disease has developed, you would resort to uh, treatments that are controlling inflammation. But the issue is that most of the clinical markers that are used to check for whether disease has developed or disease has progressed, they only show changes once it has affected those different cellular layers in a way that has damaged their structures. So I feel that the disease may have progressed to a later stage by the time just immune response control or inflammation may not be the only options that may work at that stage. So right now there's also a disconnect in understanding the early stages. So that might help us in connecting just the structural changes with the clinical markers that are actually used in these patients.
Do you think there is a way that somehow facilitate this inflammation to cause the repair and the kidney is healthy afterwards, or it's better to just inhibit the inflammation?
I didn't get that. So facilitate.
Yeah, if you facilitate inflammation to be successful and repair the cells, then you won't have this problem anymore, right? But if you inhibit inflammation, they will be good for some time, but the disease is going to be there still, right?
Right.
So which approach do you think is more feasible or better to do?
I mean, I think again, what I understand is it depends on the stage, and just controlling inflammation or controlling one target won't be enough to control the changes in the cellular structures in the later stage.
Great talk. I have a question. So, does the model consider the change in inflammation during disease progression?
During disease progression? No. Currently, it's only looking at a fixed, like a short period of time. So 48 hours is how long the experiment was conducted, the in vitro experiment. So we're only looking for that shorter period of time of 48 hours. But if we do, like the last slide that I showed, if we do look at the effect of these mediators in diabetic mice, we may be able to capture the changes at a longer time scale. Another limitation is that we are not considering anti-inflammatory mediators currently. So we may also need to include those to see how inflammation changes over time.
Take the chair's prerogative to ask a question. It seemed like both your training data sets from the literature and validation, a lot of them, they have just one or two, maybe
Yeah.
3 data points, but one of them is 0, 0. Are they really doing time-dependent measurements, or is it sort of like a single point measurement?
It is, so most of those are a single point measured at 48 hours or at the end of the experiment. And the first point is usually just what was measured in a healthy case.
All right, let's thank Krutika again.